Hello children, welcome to Vivid Botany. Today we have another question about the anatomy. So we have been given here the points. All the points indicate the one and only organ of the plant, that is monocot stem. TS of monocot stem. So here I will explain the features of monocot stem briefly. So in monocot stem, the external feature, that is outermost layer, it is called as epidermis. This epidermis is uniseriated. Cells are compact, no intercellular spaces, leaving parenchymatous, and there may be stomata present in between them, meant for transpiration and gaseous exchange, and no trichomes are present in monocot stem. Then below this epidermis, we find here the sclerenchymatous hypodermis. This is sclerenchymatous hypodermis is present beneath the epidermis. It provides hardness, rigidity, mechanical support to the stem. As monocots do not have the secondary growth, the hypodermis, sclerenchymatous hypodermis provides take care of its mechanical support, hardness and rigidity. So this is called as sclerenchymatous hypodermis. Then next to sclerenchymatous hypodermis, we find all this is the ground tissue. So in this ground tissue, there is no difference of the endodermis, pericycle, medulla or pith, medullary rays, all that are here absent. So all that are absent here. Let me write what are absent in monocot stem. Number one, there is no cortex. Cortex is absent, and there is no stele. Stele is also absent. Then no medulla. Medulla or pith, that is absent. Right. And endodermis is also absent. Pericycle is also absent. Then one may question why these are all absent. The one and only reason is that here all the vascular bundles in monocot stem are scattered. They are not arranged like a ring as we see in case of the dicot stem. So here all the vascular bundles are here scattered. So how they are scattered? In what manner they are present? Usually they are numerous. They are not 1, 2 or 10. They are many in number. They are numerous and they are not of same size. Here the peripheral vascular bundles, I write VB, are smaller and many in number, whereas the central vascular bundles are fewer and larger in size. So this you should remember. Peripheral vascular bundles are many in number. These are many in number. Whereas the central vascular bundles are less in number, few in number, but larger in size. That is the difference you should remember here. Now, if you see each vascular bundle that is enclosed by sclerenchymatous fibers, so they are called as fibro vascular bundles. Why they are called fibrovascular bundles? Because they are enclosed by sclerenchymatous fibers. Now I am drawing the structure of one vascular bundle. So usually this is one vascular bundle. Here we find the metaxylem. Metaxylem means primary xylem only, but later formed xylem is called as metaxylem, and this is the protoxylem, and this is the protoxylem lacunae. And here you find the phloem. This is phloem. And here around this you find here the sclerenchymatous bundle sheath cells. Though this is a typical monocot stem vascular bundle. It is enclosed by sclerenchymatous fibers. So this is sclerenchymatous fiber. So they are called as fibrovascular bundles. It is phloem. This is metaxylem. And this is protoxylem. And it is protoxylem lacunae. Protoxylem lacunae. Right. How do we understand this protoxylem lacunae? Actually, what happens here, protoxylem, metaxylem both are primary xylem only, but this is later formed primary xylem. Later formed primary xylem. This is first formed primary xylem. First formed primary xylem. You know that protoxylem is inwards, metaxylem is outwards. So we call it as end arc. Protoxylem is inwards, metaxylem is outwards. How do you identify the protoxylem and metaxylem? Remember, protoxylem is small in size. It is formed when the plant is very young. Metaxylem is large in size. It is formed when the plant is grown up. So generally, protoxylem whales are small in size. Metaxylem whales are large in size. So based on the arrangement of protoxylem and metaxylem, the two types of xylem can be found, that is exarch and endarch. Generally in stems, protoxylem is inwards, metaxylem is outwards. Such cases it is called as endarch. Endarch is common function feature of the stem, may it be dicot stem or monocot stem. And in roots what happens generally, protoxylem is outwards, metaxylem is inwards. Such is called as exarch. And what is this protoxylem lacunae? Do remember, protoxylem lacunae is formed due to rapid growth of the stem. I write it here. Due to rapid growth of the stem. Why does stem shows rapid growth in monocots? Remember, monocots do not show the secondary growth. They show only the primary growth. So within 3 months or 4 months, they complete their entire life cycle. So their growth is very rapid. When growth is very rapid, they require much water for their metabolism, for their growth and development. In such cases, what happens? That much water is passed through very forcibly through the protoxylem vessels. So that forcibly passing down water will rupture the walls of the protoxylem. As a result, protoxylem walls are ruptured and protoxylem lacunae means cavity is formed. So due to rapid growth of the stem, protoxylem lacunae is formed. This protoxylem lacunae is an example for lysigenous cavity. Lysogenous cavity means the cavity that is formed by rupture of the cells is called lysogenous cavity. So, having protoxylem lacunae is an important feature of characteristic feature of the monocot stem. And you see here, phloem is outside. I am writing here, phloem is outside and xylem is inside. So, that is called as endoxylem, and the vascular bundle is called as conjoined. Conjoined means xylem phloem are present on same radius, and closed vascular bundle. Closed means there is no cambium. No, there is no cambium. What is cambium? The meristem, embryonic meristem is called as cambium, that is absent in monocot stem. Why cambium is absent in monocot stem? Because monocot stem does not show secondary growth. So when there is no cambium, there is no secondary growth. When there is no cambium, it is closed type of vascular bundle, right? So conjoined closed vascular bundle with endark xylem, and here water also absent in monocot stem, that is phloem parenchyma. Phloem parenchyma is also absent in monocot stem. Phloem fibers, because phloem fibers are part of secondary phloem. As there is no secondary growth, there is no secondary phloem in monocot stem. So as there is no secondary phloem, there are no phloem fibers in the monocot stem, and there is no secondary growth also. Secondary growth is also absent. Now it is very clear, I think, that water absent in monocot stem. Cortex, stele, medulla, endodermis, pericycle, phloem parenchyma, phloem fibers, secondary growth, medullary rays are also absent in monocot stem. And as the scattered numerous vascular bundles are present in the monocot stem, it is called as atactostele. It is called as atactostele. In case of dicot stem, we call it as eustele. Eustele means true stele. In case of monocot stem, we call it as atactostele. So this is the explanation of the monocot stem. Now let us see the question here. That is, the transverse section of a plant shows following anatomical features: large number of scattered vascular bundles surrounded by bundle sheath. This is sclerenchymatous bundle sheath. You know, here sclerenchyma is present in the form of hypodermis as well as the bundle sheath cells. A large conspicuous parenchymatous ground tissue. Yes, because there is no differentiation, demarcation of the hypodermis. I mean, hypodermis is there, of course, but thereafter cortex, endodermis, pericycle, stele, medulla, medullary rays, all are here absent. Instead, the ground tissue is present in between that. So this is the ground tissue in which we find these vascular bundles. So vascular bundles are scattered in monocot stem. So if you have fields, you can go and cut the TS of maize. You know maize, that we eat maize, corn during the rainy season. So if you cut the TS of corn, you will find here that in the parenchymatous, that is chlorenchymatous ground tissue, scattered vascular bundles are found called atactostele. They are usually peripheral or smaller, many in number, and the central are larger in size and few in number. So the second one. Next we see vascular bundles are conjoined and closed. I have told you here what is conjoined, what is closed. Conjoined means xylem phloem present on same radius. Closed means no cambium. I write here xylem phloem on same radius, right? And next, phloem parenchyma absent. I told you that. Identify the category of the plant and its part, that is monocotyledonous stem. So it is the answer for this question. So really very beautiful question. Generally they give characters and they find out what it is. Generally they give the organs and they ask the features of that organ. For example, if we dicot stem, what are the features of dicot stem? In dicot stem we find cholenchymatous hypodermis, parenchymatous general cortex, what is that? Starch sheath as endodermis, sclerenchymatous pericycle, regularly arranged vascular bundles. They are 15 to 20, arranged in a ring-like manner, called as U-stele, right? And medulla is present. Medulla extends in between vascular bundles called as medullary ray. They are features of the dicot stem. If you see the dicot root features will be including here that it is diarch to tetrarch. Diarch to tetrarch means 2 to 4 vascular bundles are present. And exarch. Exarch means protoxylem is outwards, metaxylem is inwards. And pith is inconspicuous, that is very, very scanty, narrow due to extension of metaxylem wheels towards the center. That is dicot. Coming to monocot root. Dicot, monocot, both are almost same. Then what are the differences in between dicot root and monocot root? In monocot root it is polyarch. Polyarch means vascular bundles are more than 6. Right? Exarch. Exarch is common. But in monocot root, the pith is extensive, very big pith that we found. The medulla is very big in case of monocot root. And further, in monocot root, there is no secondary growth. Secondary growth is absent in monocot root. In dicot root, secondary growth is seen, and secondary meristem is formed from parts of pericycle, conjunctive tissue, as well as lower part of the phloem it is formed. So these are the features of the dicot root, monocot root and dicot stem. So generally what they do, they give what are the features of monocot root, find out the features of dicot root, give the odd one about the dicot stem. Like that they give these, they give the root or stem and they find you to find out the features of that root or stem. So what I suggest you is that you better make a chart of 2 roots and 2 stems. Of course 2 leaves are also there. Dicot root, dicot stem, monocot root, monocot stem. Write them on a paper. Better if you draw a diagram, it is very good for you. Then read those points every day. Unless and until you get, don't leave that. Once you get the points, once you understand them, once you memorize them, then you can identify easily, you can remember them and you can do this kind of bits without any difficulty. So hope my explanation is helpful for you. It will make you better for the preparation of botany as far as our NEET is concerned. So you have any doubt, please drop a line and make a comment, and please show your response in the comment. I am talking from the Vivid Botany. This channel is aimed at clarifying and making ease of botany at the NEET UG entrance, right? So hope you are enjoying this video, you are learning from this video. Remember, we are not wasting our time, and I expect you also not wasting of time, and do prepare well and do hard work and knock the door of medical college as early as possible. The exam is conducted. Hope you are doing well. Thank you very much, and subscribe my channel Vivid Botany.